Hi, my name is Chiaramello. I'm an associate professor of anatomy and cell biology and the director of the Mitochondrial program at the George Washington University School of Medicine and Health Sciences. Today, I will discuss with you the survey analysis of of patient input on the clinical spectrum of LH1 plus. It is an ongoing study with the LH1 plus global project to accelerate discovery into patient care. Before I present our key data, I want just to acknowledge the key contributors of that study. Lisa Thompson, who is the founder of the LH1 Plus Global Project and LHON Canada, and she has spearheaded this survey project. Then my longtime collaborator, Dr. Andrea Goldman, who is a professor of neurology and pediatrics at Children's National Medical Center. Dr. Martin Newton-Bogard, who is a lead research scientist in my laboratory, and Eliana Goldman, who is an undergraduate student at the University of Michigan. She has been doing her summer research for the last 2 years with us. Those studies were funded by the Department of Defense Discovery Award. So I want to spend a little bit of time on describing the disease LH1 plus, since there's a lot of misconception and confusion in the nomenclature between the LH1, LH1 plus, and LH1MS. So first of all, the LH1 plus disease is a maternally inherited progressive neurodegenerative disease with an adult onset. What does it mean to be maternally inherited? It means this is a disease that is caused by pathogenic mitochondrial variants mapping in a mitochondrial genome. Since the mitochondrial genome is solely inherited from the mother, then this disease is maternally inherited. There are two key features that distinguish the LH1 patient from the LH1 plus patient. The LH1 plus disease has a gender bias with a female predominance. That stands in contrast with the LH1 disease that has a male predominance. Second, most LH1 plus patients have vision impairment, but not all. You do have some LH1 plus patients without any vision loss or vision impairment. But what they all have in common, the LH1 plus patients, is that they all have a spectrum of ocular symptoms. The problem is that those extraocular symptoms appear later than the vision loss. So vision loss is usually the very first when it occurs. It's usually the very first symptom just to show up. And this is what prompted usually the initial diagnosis, clinical diagnosis of LHON. And then when the additional extraocular symptoms start appearing, then there was never any reconsideration on the diagnosis. So you do have a lot of patients diagnosed with LH1 when in fact they are LH1 plus. So the presence of a spectrum of extraocular symptoms makes the definition of LH1 plus. So what are those? Below is a list of the most prevalent extraocular symptoms. It's by no means an exhaustive list. So the first one is multiple sclerosis-like presentation, exercise intolerance, mental and physical fatigue, migraine, peripheral neuropathy, tremors, anxiety, cardiac arrhythmia, myopathy, dystonia, bladder spasm, urinary dysfunction, and gastrointestinal dysmotility. The most common, the extraocular symptoms that they all share are exercise intolerance, mental and physical fatigue, anxiety, even in the LH1 plus patient without vision loss. So we cannot say that the anxiety is due to vision loss. And then you have like a mixed bag of extraocular symptoms among all the patients. So you can have patients with just one of them, migraine, and others with a combination of a spectrum of extraocular symptoms. So I believe that now it has been clarified and kind of like proven that the LH1 plus disease is a clinical entity in itself that should be separate from the LH1 disease. As for the LHON-MS, which stands for LHON, LHON multiple sclerosis-like illness, is usually the patients that have vision impairment or not, but essentially have MS-like presentation. So it's a small subset of LH1 plus patients. And the frequency of the LH1 MS is much lower than the frequency of LH1 plus. The next slide shows the demographics of our de-identified survey using the digital platform of LH1 Plus Global Project. The schematic on the left shows the gender distribution among survey participants that really confirm the female predominance among the LH1 Plus patients. We had 65% of the survey participants being female and only 35% of them being male. The graph on the right shows the age distribution of the survey participants. And the majority of the survey participants were 22 years old or older, as expected, since it's essentially an adult onset disease. However, our survey revealed a small subset of participants or patients that are much younger. We found 10 patients that were between 13 and 21 years old, and then 8 patients that were less than 12 years old. So that was really interesting because it was never reported until now. It was LH1 plus disease was always thought just to be like an adult disease, but apparently it can occur in children. And I'll come back to this subset of patients on my very last slide. So then we asked the participant whether they had at least one family member with LHON or multiple sclerosis. And the schematic on the right indicate that more than 60% of the survey participants answered yes to the question of family history of LHON. Contrast this with less than 20% of patients answering yes to the question of family history of multiple sclerosis-like illness. And that kind of makes sense given the low prevalence of LH1MS. The next slide illustrates the problem that we're having right now is that most patients with LH1 plus symptoms receive an LHOS diagnosis even though they have symptoms specific to LH1 plus. And that is shown on the graph on the left, showing that there are 68% of the survey participants and that received an LH1 diagnosis even though they have LH1 plus symptoms. And only 25% of the survey participants received an accurate diagnosis of LH1 plus. And finally, as expected, less than 10% of the survey participants receive a diagnosis of LH1 MS. So bottom line, there's a lot of patients that are misdiagnosed. So then we wanted just to find out how the LH1 plus patients are essentially diagnosed. And the survey revealed that the majority of them, about 70% of them, were diagnosed initially by an ophthalmological evaluation that was prompted upon vision loss or vision impairment. Usually when this occurred, the patient is referred to a neuro-ophthalmologist that confirm the LH1 diagnosis. But the neuro-ophthalmologist is never going to look the patient as a whole, and then all those extraocular symptoms are not taken into account. The next test that was previously administered was brain MRI, and finally blood test just to get a genetic diagnosis. Below are two graphs pertaining to the muscle biopsy on the left and the skin biopsy on the right. The muscle biopsy on the left shows that they were like about 15% of the survey patients that underwent the muscle biopsy. I'm not surprised by this very low number of 15%, since the muscle biopsy is a highly invasive procedure that requires a general anesthesia, and patients with a mitochondrial disease usually don't feel that well with some general anesthesia, so it's very hard on the patient, takes time to recover, and is quite expensive as well. So it's less and less practice. It used to be the golden standard, but it's no longer. But what really perturbed me is that only 10% of the survey patient got a skin biopsy. And this is what I call a missed, because I think that a skin biopsy would actually benefit, just to have more targeted treatment. Why that? Because first of all, skin biopsy is done in a doctor's office, is not painful, it's done using an anesthetic cream, so it doesn't require general anesthesia or local anesthesia. It's done very quickly, and then it allows us just to derive patient cells and study those cells in terms of the mitochondrial deficit specific to this patient and the patient's mitochondrial metabolic signatures. So when we combine all this data along with a genetic result with next gen sequencing approach, then we have like a comprehensive overview of the pathological events occurring in this patient, and that will really favor more like a personalized medicine than rather than you know um, standardized treatment. So this is definitely a missed opportunity. So why is it so difficult for those patients to receive the appropriate diagnosis of LHON plus? It's because the phenotypic manifestation of this disease are highly heterogeneous among patients and even among members of the same family. But so that means that those extraocular symptoms vary from patient to patient and family member from member within the family, and that we don't know the mechanism behind. So the graph on the left shows that about 60% of the survey participants answered yes to vision loss, but you can see that there's about 30% of them that said no. So that pool of 30% again didn't fit the profile of LHON because they didn't have vision loss, and maybe as the extraocular symptoms started showing up later in life, since it's a progressive disease, maybe that led to the proper diagnosis of LH1 plus. And two prevalent extraocular symptoms we noticed from the survey were the muscle weakness and the hand tremor. And as expected, only less than 10% of the survey participants had seizures. Then we asked them this unusual question whether those patients experienced bladder spasm and had urinary retention dysfunction. And 30% of them answered yes to this question. And in fact, they were even surprised that we asked this question since nobody even bothered inquiring why they had bladder spasm or urinary retention dysfunction. So this is a very much underreported and unattended clinical symptom. So in terms of the pathogenic mutation or mitochondrial variants responsible for causing LH1 plus, there are seven known ones. And I've grouped them into two categories, the most frequent category and the least frequent category. Among the most frequent category, you have the 11778 G2A that is present in 40% of the survey participants, and this is the one that is in common with the LHON patient, hence the confusion between LHON and LH1 plus. The second most prevalent mitochondrial variant is a 14484 T2C, and the third one is 3460 G2A. And there are four least frequent mitochondrial variants that make less than 1% each. The next slide will show how interesting, in fact, they are, because our survey revealed an unexpected finding is that three of those four rare variants shown here in the blue box, the 14459 G2A, the 15512 T2C, and the 14258 G2A, they actually cause a childhood onset of LH1 plus. So it's an onset that occurs between the age of 5 and the age of 15. So definitely those three mitochondrial variants have a different penetrance than for example, the 1177A to G2A. So there's a different mechanism. So those will be very interesting to study. And that highlights the fact that the misconception that the LH1 plus is an adult disease, when in fact it can be found in a pediatric patient population. So in summary, the LH1 plus disease is a clinical entity in itself that deserves its own criteria and its own diagnostic criteria. Second, there's a lot of phenotypic heterogeneity associated with this disease. And third, the predominant onset is adult onset, but it doesn't exclude there are cases of childhood onset. So I want to thank you for your attention. I'll be happy to answer any question. If you want to visit our website, here's a hyperlink for further information on our study. Thank you.